Welcome. Today, we're diving into the world of stability indicating assays, focusing on solid oral dosage forms. I'm Dr. S.K. Singh, and together, we'll explore how these assays help ensure the safety and effectiveness of medications over time. Let's get started. Let's start by understanding what a stability indicating assay, or SIA, really is. It's a precise, validated method that measures the active ingredient in a drug, even when other substances like degradation products or impurities are present. This is crucial because it helps determine how long a product stays effective, supports stability studies, and reveals how the drug might break down over time. Ultimately, it ensures quality, safety, and meets regulatory demands. SIAs apply to a range of products, from tablets and capsules to powders and modified release forms. Let's take a look at the regulatory framework that guides our work. We follow several key ICH guidelines, covering everything from stability and photostability testing to impurities and analytical method validation. Alongside these, pharmacopoeial references like the USB and European and British pharmacopoeias provide essential standards. The main goal here is clear: to prove that our analytical methods can reliably separate and measure the active pharmaceutical ingredient and any degradation products throughout the entire product lifecycle. When we talk about stability indicating methods, there are a few essential qualities they need to have. First, specificity. They must clearly tell the active ingredient apart from any breakdown products. Then, accuracy, making sure we measure the active ingredient correctly every time. Precision is key too, meaning consistent results under normal conditions. Linearity ensures the method responds reliably across different concentrations. Robustness means small changes in the process won't throw off the results. And finally, sensitivity, the ability to detect even tiny amounts of degradation. All these together build trust in the data. When we talk about degradation in solid oral dosage forms, there are several key mechanisms at play. Hydrolysis happens when moisture gets involved, breaking down the compound. Oxidation is a reaction with oxygen or peroxides, which can alter the drug's structure. Photolysis occurs under exposure to UV or visible light, causing breakdown. Then there's thermal degradation, triggered by elevated temperatures. Demidation is common in peptide-based products, while isomerization involves structural rearrangement of the active ingredient. Our goal here is clear: to identify all these pathways and develop a method that can detect every degradation product reliably. When we talk about forced degradation studies, we're looking at how substances break down under different conditions. This table shows the typical treatments used: acid and base hydrolysis with mild concentrations, oxidation using hydrogen peroxide, heat between 60 and 80 degrees, UV light exposure, and controlled humidity. These stress tests help us understand how a compound degrades and ensure our methods can specifically detect those changes. It's all about making sure our analysis is both reliable and precise. Let's dive into some additional techniques used alongside HPLC. These include UPLC, LC-MS-MS, HPTLC, and capillary electrophoresis. But HPLC remains the primary stability indicating tool. Why? Because it offers high specificity and excellent resolution. It's quantitative, profiles impurities effectively, and is widely accepted by regulators. Typical HPLC conditions involve a C18 column, UV or PDA detectors, gradient elution, and a buffered mobile phase, all carefully chosen to ensure reliable, reproducible results. Here, we see a detailed example of how an assay calculation is performed using HPLC, or high-performance liquid chromatography. We start with preparing the standard 100 milligrams of the active pharmaceutical ingredient diluted to 100 milliliters. The sample preparation mirrors this, using tablet powder equivalent to the same amount of API, also diluted to 100 milliliters. The table compares the standard and sample areas, showing values close to 850,000 and 830,000 respectively. These results form the basis for determining the assay value. Let's walk through an example of degradation calculation. This helps us understand how a system's performance changes over time or under stress. By breaking down the numbers step by step, we can see where losses happen and how they add up. This example makes the process clear, showing us what to expect and how to plan for it. It's a practical way to turn theory into something we can measure and manage. Now, let's turn to mass balance assessment. This is all about tracking the flow of materials in and out of a system. By measuring inputs, outputs, and accumulation, we get a clear picture of how everything adds up. It's a fundamental tool for understanding processes, spotting inefficiencies, and ensuring nothing gets lost along the way. Mastering mass balance helps us make smarter decisions and optimize performance. Now, let's focus on validating stability indicating assays. These assays are crucial because they help us detect changes in a product's quality over time. Validation ensures the assay is reliable, accurate, and specific to the product's degradation. Without this step, we can't confidently track stability or guarantee safety. So this process is about proving the assay truly reflects what's happening as the product ages, no shortcuts, just solid science. Here, we're looking at how the product holds up over time. The key point is that it stays within the set specifications, which means it's performing as expected. The stability profile is solid. The assay starts at 100% initially, then gently declines to 98.6% at 3 months and 97.2% at 6 months. This slow, steady change shows the product remains stable and reliable throughout the study period. Let's take a closer look at a real-world example, an immediate release tablet. This case study shows how design choices impact how quickly a medication works. We'll explore the formulation, the challenges faced, and the solutions that made it effective. It's a clear illustration of turning theory into practice, and the results speak for themselves. When developing analytical methods, we often face a set of common challenges. Co-eluting degradation products can overlap with the main API peak, making it hard to separate them clearly. Detecting low-level impurities demands highly sensitive techniques. Matrix effects, like interference from excipients, complicate the analysis further. Assessing peak purity usually calls for advanced tools like PDA or LC-MS. To tackle these issues, we can optimize gradients, try alternative columns, adjust pH levels, or use orthogonal analytical techniques to get a clearer, more reliable result. We rely on stability indicating assays to guarantee that medicines stay safe and effective from production to use. These tests confirm a drug's identity, strength, and purity over time, meeting strict global standards. To succeed with regulators, we need clear specificity, thorough mass balance, validated methods, and ongoing stability checks. These assays are vital for determining shelf life, supporting stability studies, and managing the entire product life cycle. Developing them means rigorous degradation testing, strong separation techniques, full validation, and precise impurity analysis. Thanks for joining me today. If you want to explore this topic further or have any questions, please don't hesitate to reach out. I appreciate your time and attention, and I look forward to continuing the conversation. Thank you.